Dr. Sage here. In the second part of Chapter 14, we're going to cover the things that Mendel did not discover. While Mendel did discover a lot about genetics, he didn't see everything in regards to genetics. But before we get into those details, one thing I wanted to make note of is that there's a common misassumption that many people have. Most people assume that the dominant allele is the one you will see more often in the population. It's dominant, so it must be the one you see most often. However, that would be an incorrect assumption. For example, there's a condition called polydactyly, which is caused by a dominant allele. When somebody has polydactyly, what happens is they're born with more than 10 fingers or more than 10 toes. However, even though this is a dominant trait or dominant allele, most babies are not born with 11 fingers. So just because something is dominant does not necessarily mean you see it more often within the population. So get rid of that misassumption. Now, Mendel, when he was doing his studies, he studied a lot about genetics, but he didn't see everything. For example, when Mendel was doing his studies, he saw what's called complete dominance. So purple is completely dominant over white. If you have a purple allele and a white allele, you see the phenotype of the purple allele, you don't see the white allele. But there are different types of dominance. Also, when Mendel was doing his experiments, the genes he looked at only had two alleles, like purple or white flowers, yellow or green peas, round or wrinkled peas. But a lot of genes has more than two alleles. And another thing that Mendel did not see is that a gene can't produce more than one phenotype. So we're going to start by discussing these things that Mendel did not observe. So first, there's different types of dominance. What Mendel saw is called complete dominance. So in complete dominance, the homozygous dominant and the heterozygous look exactly the same. You cannot tell them apart. The dominant purple is completely dominant over the recessive white. That's complete dominance. But there are different types of dominance. For example, besides complete dominance, you can have incomplete dominance or codominance. So, the example I'm going to give you for incomplete dominance is again using flowers, but this time instead of the flowers of pea plants, it's going to be the flowers of snapdragons. So in snapdragons, if you take a white flowered snapdragon and made it to a red flowered snapdragon, you do not get white flowers or red flowers. What you actually get is pink flowers. So what this kind of looks like is it kind of looks like blending. Like, for example, if you take red paint and white paint and mix them together, what you'll get is pink paint. Like the two colors are blended together. However, this is not blending, because if you take pink paint and mix them together, from that you'll never get red paint or white paint. You'll always get pink paint. But if you take pink flowers and mate them with pink flowers, you'll get pink flowers and white flowers and red flowers. So what's actually happening here is the homozygous dominant makes red flowers. The homozygous recessive makes white flowers. And the heterozygote makes pink flowers. So this is called incomplete dominance. With incomplete dominance, the heterozygote, its phenotype is halfway between the two homozygotes. So pink is halfway between red and white. The heterozygote, its phenotype looks halfway between the two different homozygotes, homozygous dominant and homozygous recessive. That's what incomplete dominance is. Another type of dominance is called codominance. This is where two alleles are expressed equally and separately. One is not dominant over the other. They are codominant. Now, the most common way you'll hear this referred to is with the human ABO blood groups. So we need to learn about the human ABO blood groups. First thing you need to know is that in humans, you can be either blood type A, or you can be blood type B, or you can be blood type O, or you can be blood type AB. So those are the human blood types. We're only talking about the ABO blood groups. If you know blood type, we're not talking about the Rh factor, the positive, negative, or the Mn factors, only the ABOs. Okay, well, these blood types are really the phenotypes, and they have genotypes that go along with them. So, for example, blood type A, okay, what that can be, genotype, is either AA or AO. Okay, so A is dominant over O. Blood type B can be either BB or BO. So B is dominant over O. Blood type O can only be OO. So that's the recessive. So far this looks like complete dominance. The reason it is not complete dominance, the reason it is codominance, is because of the last blood type AB. Blood type AB, its genotype is AB. A is not dominant over B. B is not dominant over A. They are codominant. They're expressed equally and separately. Okay, so to help you understand a little bit more about what that means, the human blood types, what it's really doing is it's telling you what type of carbohydrates are stuck in the outside of your red blood cells. So, for example, if you're blood type A, you have A-type carbohydrates stuck in the outside of your red blood cells. Whereas if you're blood type B, you have B-type carbohydrates stuck on the outside of your red blood cells. If you're blood type O, you don't have any of these carbohydrates stuck on the outside of your red blood cells. And if you're blood type AB, you have both A-type and B-type on the outside of your red blood cells. They're expressed equally and separately. A is not dominant over B. B is not dominant over A. They are codominant. Okay, so that's what the human blood types are really telling you. All right, now with the human blood types, for the exam, you have to have these blood types memorized. Like you need to know blood type A can be either genotype AA or AO. Blood type B can either be BB or BO. O is OO, and AB is AB. Okay, but there's also a more formal way of writing it. So you also have to know the other way of writing it. So more formally, genotype AA, the way you would technically write it is uppercase I, superscript A, uppercase I, superscript A. That means AA. AO would be uppercase I, superscript A, little I. Okay, so lowercase I means O. Genotype BB would be uppercase I, superscript B, uppercase I, superscript B. BO would be uppercase I, superscript B, lowercase I. So that's BO. OO would just be little I, and AB would be uppercase I, superscript A, uppercase I, superscript B. Okay, so that's a more formal way of writing it. Okay, what's displayed on this screen here, this you have to have memorized for the exam. It's not going to be provided for you. Okay, so make sure you learn that. Now, the reason you need to know the human blood types is because there's going to be genetics questions on the exam you have to solve with human blood types. For example, this is an example genetics problem that relates to human blood types. So let's say there's a woman that's blood type O. Her child is also blood type O, and ask what are the possible genotypes of the father. Now, for human blood types, you might draw out a Punnett square to solve them, or you might not. But anytime I have a genetics problem, the first thing I do is I read the entire problem, and then I go back and I reread it, and I start drawing out what I'm reading. Okay, so I've read the whole thing, now I go back, and I see that there's a woman, okay, who is blood type O. Okay, so she's blood type O. And she's obviously going to mate with some guy. They're going to have a child who's blood type O. So the kid is going to be blood type O. And then the question that's asking you is, what are the possible genotypes of the father? So what are the genotypes that the father can be? All right, so all I've really done is I just drew out what this question was actually saying. But by doing that, it better helps me to understand the question. So, for example, if the woman is blood type O, based upon what we will have memorized, if she's blood type O, we know she has to be OO. Okay, so her genotype would be OO. Her kid is also blood type O, which means the kid also has to be genotype OO. Now, the mom has to give her kid something. all she has is an O, which means one of the O's that the kid has came from their mom, which means the other O that the kid had has to come from their dad. So their dad has to have at least one O. Okay, so what that means is the possible genotypes of the father, what the dad can be, is anything that has at least one O. So if we go back to this table here, what the dad can be is dad can be blood type A, because he can be AO, or blood type B, because he can be BO, or he can be blood type O, because he can be OO. So the dad can be AO, or BO, or OO. He can be any of those blood types. The one blood type he cannot possibly be is blood type AB, because blood type AB doesn't have an O, so he can't be blood type AB. All right, so that's how I would solve that genetics problem. And if you notice, I didn't do a Punnett square. I just read through the word problem and then drew out the word problem and figured it out from there. Okay, so here's another one. Now, what I'd like you to do is make sure you have your pencil and paper again, and I'd like you to pause the video now, and you work through this problem and try to solve it. Make sure you try to do this on your own, and then when you think you have the answer, unpause the video, we'll come back, and I'll work through it with you. But try to do it on your own first. Okay, so I read through the problem. A newborn infant is type A, the mom is type O. What are the possible genotypes of the father? Okay, now I go back through, I reread it, and I draw one reading. Okay, a newborn infant is type A. Okay, so the baby is going to be blood type A. Okay, the mother, it says the mother is blood type O. And then it asks, what are the possible genotypes of the father? Okay, so with that information, now I just drew out what I was reading, but now I can start filling in some of the information. For example, if the mother is blood type O, she has to be OO, because the only way to be blood type O, since that's the recessive, is if you're OO. Now the mom has to give her kid something. The mom could not have given her kid the A, because the mom doesn't have an A, which means the mom had to pass on an O to her kid, so the kid has to be AO. Which also means, since the O came from the mom, the A had to come from the dad. So the dad is anything that has at least one A. Okay, so the dad can be genotype AA or AO, or one that people forget about a lot, he can be blood type AB, because that does have an A. All right, so that would be the answer. These are the possible genotypes of the father. So when Mendel was doing his experiments, the genes he looked at, there were only two possible alleles. For example, the pea plants had either purple flowers or white flowers. There was no blue flowered or yellow flowered or orange flowered or red flowered plants. There were only two possible options. But it turns out that a lot of genes have more than two possible alleles. For example, the gene that controls coat color in rabbits. It's not just lowercase c or uppercase c. You can have lowercase c, or you can have ch, or you can have cch, or you could have capital C. Okay, now you don't need to memorize these. The point of this is when you have more than two alleles, you can make more possible phenotypes. So the more alleles you have, the more possible phenotypes you can make. The next concept is a term called pleiotropy. And pleiotropy means that one gene creates more than one phenotype. So the example I'm going to give you for that is Siamese cats. If you've ever seen a Siamese cat, you know they have a very distinct pattern to their fur. But if you ever really looked at a Siamese cat, you'll also realize that they always have crossed eyes. So these two very different phenotypes, the pattern of their fur and their eyes being crossed, are actually caused by the same gene. Okay, so that's called pleiotropy, when one gene creates more than one phenotype. All right, so for the next term we have epistasis. Epistasis is the situation where the phenotypic expression of one gene depends on a different gene. The example I'm going to give you for epistasis involves the dogs Labrador Retrievers. If you ever seen a lab before, you know you can have a black lab, or you can have a chocolate lab, or you can have a yellow lab. Okay, now it turns out that there's a gene in Labrador Retrievers that actually makes the pigment, the color that goes into the fur. So to be a black lab, black is the dominant. So to be a black lab, you have to have at least one uppercase B. Okay, it doesn't matter what the other B is. It could be big B, or big B, little b, and you get a black lab. To have a brown lab, that's the recessive, you have two lowercase b's. Then you'll make the brown pigment, which will go into the fur. Okay, well, how about a yellow lab? All right, well, this gene here, the B gene, actually makes the pigment, like the actual black color or brown color that's going to go into the fur. But it turns out there's a second gene, which we're going to call the E gene, which takes this pigment and actually puts it into the fur. So to be a black lab, you have to have at least one uppercase B to make the black pigment. And you have to have at least one uppercase E to take that black pigment and put it into the fur. To be a chocolate lab, you have to have two little b's to make the brown pigment and at least one big E to put that brown pigment into the fur. Now, a yellow lab, what they have is they have two little e's. Okay, it doesn't matter what pigment they're making because they're not actually putting it into the fur. That makes a yellow lab. Okay, so black lab, at least one big B makes the black pigment, and at least one big E puts the pigment into the fur. Chocolate lab, two little B's makes the brown pigment, at least one big E puts the pigment into the fur. Yellow lab, okay, has two little E's. It doesn't matter what pigment it's making. It could be making black pigment or brown pigment, but since it's not putting it into the fur, you can't see it. So this is called epistasis. The phenotypic expression of one gene, the B gene, depends on another gene, the E gene. Okay, so that's epistasis. Now, when Mendel was looking at his experiments, for all the characters of genes he looked at, there was two options. Like it was either purple flower plants or white flower plants. That was it. It's not like you had a really deep purple, then kind of a medium purple, then a pale purple, then kind of a purplish white, and then a white. He didn't have a continuous range of colors. It was one or the other. But for a lot of genes, we do have a continuous range. So for example, human height, for example. As humans, we are not all either exactly 5 foot tall or exactly 6 foot tall. We have a continuous range of human heights. Okay, so quantitative characters are those that vary in a population along a continuum. Quantitative variation usually indicates polygenic inheritance. Okay, so remember, poly means many, and genic means gene. So what this means is more than one gene is adding up together to give you the phenotype. So another example of that is skin color in humans. Okay, human skin color is not determined by one gene. It's determined by 6 separate genes. And which alleles you have of those 6 separate genes gives us this continuous range of human skin color that we see. So far, everything I've told you about genetics is that your genes determine your phenotype. Okay, in other words, which alleles you have determine what you're going to look like. But it turns out genes are not the only thing that determines your phenotype. For example, these flowers here, okay, these have the same genotype, but obviously their phenotype is very different. For these flowers, depending on whether you raise the flowers in more acidic soil or more basic soil, the flowers would come out different colors. So it turns out it's not only your genes that determine your phenotype, it's also your environment that you're in. Even the environment within an organism can affect its phenotype. For example, this rabbit here has a gene that says make black fur. And that gene is everywhere in the rabbit. Okay, it's saying make black fur. Okay, but rabbits are just like us in that our extremities, so our ears, nose, fingers, and toes are colder than our core body. Well, the same is true for the rabbit. So its core body is warmer than its extremities. And this gene that says make black fur is affected by temperature. Okay, when the gene is in colder areas like its ears and its nose and its paws, the gene can make black fur. But in the warmer areas like its core body, the gene cannot make black fur. So even the environment within an organism can determine its phenotype. Okay, so far I've said absolutely nothing about humans. Now, humans do follow the same rules of genetics as pea plants that Mendel discovered. In fact, the main difference between humans and pea plants in regards to genetics is that we don't do genetics experiments in humans for several different reasons. First, the generation time is too long. Remember, Mendel was doing experiments when it went through 3 generations, the P generation, the F1 generation, and the F2 generation. Okay, if you're a scientist and you're doing a genetics experiment on humans and you had to go through 3 generations, you would die before your experiment is done. Second, in humans, parents produce relatively few offspring. So genetics involves statistics and probabilities. And anytime you're working with statistics and probabilities, what you need is a very large sample size. For example, when Mendel was looking at his pea plants, he didn't look at like 4 baby pea plants. He looked at like 1,000 baby pea plants. Well, a human couple is not going to produce 1,000 offspring. So humans don't make enough offspring to get the large sample sizes you need to work with genetics. And the third reason, actually main reason that we don't do genetics in humans is because a lot of genetics experiments would be unacceptable or unethical to do in humans. For example, I can easily take one pea plant and tell it has to mate with another pea plant, or one fruit fly and say you have to mate with this other fruit fly. And then you're going to have baby fruit flies and you're going to do experiments on your babies. Okay, I can easily do that with a fruit fly. If I try to do that with a human, they throw me in jail. Okay, you can't do that. Okay, so we just don't do genetics experiments in humans. So instead of doing genetics experiments, what we do is we look at essentially what's already been done. In other words, we look at the family histories of people or their family trees. And there's a particular way of diagramming family trees in genetics, which is known as a pedigree. Okay, and you have to be able to read and understand pedigrees for the exam. So a pedigree is basically just a family tree, but it traces a particular genetic disease, or it doesn't have to be a disease, it could just be a genetic trait. So this is an example of a pedigree. A pedigree is basically symbols that mean things. All right, so in a pedigree, circle means a biological female, and a square means a biological male. Okay, so that's what a circle and a square mean. Okay, a line connecting a circle and a square, that means a mating. And then what comes off of that line, those are the kids of that mating. So, for example, this couple here, they had a daughter, and then they had a son, and then they had a daughter, and then they had a son. Okay, now of note, this is not their son. Because first, if this was their son, there'd be a line connecting it, kind of like this. And second, if that was their son, then that means there's some incest going on. And incest is bad. One of the things you're going to get out of the lecture today is incest is bad. Okay, so in this pedigree, what we're doing is we're looking at 3 generations. So kind of like parents, kids, grandkids. Now, the reason we do a pedigree is to trace some genetic disease or some genetic trait. Okay, in a pedigree, if you have the disease or if you have the trait, then you're a filled in circle or square. If you do not have the disease or do not have the trait, then you're going to be an empty circle or square. Okay, so for example, he has the disease, his daughter has the disease, his son has the disease, his next daughter does not have the disease, and his next son does have the disease. Okay, so that's the basics of how you read a pedigree. Now, one of the things you have to be able to do with a pedigree is figure out people's genotype based upon the pedigree. In order to do that, one thing you have to know is what type of gene is it. So some genes, you have the disease if you have at least one copy of the dominant. So if you're big A, or big A, little a, you have the disease. Okay, so if you're little a, you don't have the disease. So that'd be a dominant disease. You have the disease if you have at least one copy of the dominant. If you don't have any copies of the dominant, then you don't have the disease. Okay, so that's called a dominant. Now, one way you can know if a disease or a gene is a dominant is, for example, in this case, it tells you. So this tells you this is dominant inheritance. So what that means is, in order to have this disease to be affected by it, you have to have at least one copy of the dominant, let's say big A, and we're going to represent it as big A dash. And that lets us keep in mind that it could be big A, or big A, little a. It doesn't really matter. You need at least one big A. If you don't have the disease, then you have to be two little a's. Because if you had a big A, then you wouldn't be unaffected. You wouldn't be an empty circular square. You'd be a filled-in circular square if you had at least one big A. All right, so let's say I want to figure out people's genotypes based upon this pedigree. What I'm going to do is I'm going to start by filling in the information that's easiest to determine. So for example, on this pedigree, since it's a dominant, what's easiest to determine is if somebody does not have the disease, since it's a dominant, that means they cannot have the dominant. So anybody that's an empty circular square, it has to be two little a's. So I'm going to go through and I'm going to put two little a's for all of the empty circle and squares. Okay, so that's like the first easiest thing to determine. Okay, the next easiest thing to determine is if somebody has the disease, that means they have at least one capital A. Now it could be big A, or big A, little a. I'm not sure which it is yet. So I'm going to go through and put big A dash for all the ones that have a filled-in circular square. Big A dash. All right, so the third and final step, which can be the most challenging step, is to figure out what the dashes are. Are these dashes big A's or little a's? Now you can figure that out by either looking at somebody's kids or by looking at somebody's parents. For example, let's say this male here, okay, this one right there, they're filled in, so they have at least one big A. Now we want to figure out what the other A is. Now he doesn't have any kids, but we can figure it out by looking at his parents. Now remember, the reason he has two copies of this gene is because one comes from dad and one comes from mom. Now the big A that we know about, we know that had to come from dad because dad has a big A. Mom can't give him a big A because mom doesn't have a big A. So big A came from dad. So the other A has to come from mom. Well, all mom has is two little a's, which means she had to pass on a little a to her son. So now we know he has to be a heterozygote. So that's by looking at somebody's parents. Now we can also do it from the other direction. So for example, let's say this male here. Okay, he's filled in, so he has at least one big A. Now let's say we want to figure out what the other A is. Now we can figure this out by looking at his kids. Okay, so how we actually can figure that out is by his daughter here. His daughter has two little a's. Okay, well, he had to give something to his daughter. The reason she has two little a's is because one little a came from her mom. The other little a had to come from her dad. Okay, so that's how we know he is also a heterozygote. Okay, so that's the basics of how you figure out pedigrees. So here's an example of a dominant trait, not a disease, a trait, which is called widow's peak. To have this hairline shape here, okay, the only way to have it is if you have a copy of the dominant. So either homozygous dominant or heterozygous. If you have this hairline shape, so not having widow's peak, the only way you can have that is if you're homozygous recessive. Okay, so here's an example of a dominant pedigree. Now, one thing to note about the dominant pedigrees is that I might tell you it's a dominant, like I did here, or you might have to figure out if it's a dominant. It might not tell you that. Okay, the way you know that it's a dominant disease or a dominant trait is if every single time somebody has it, at least one of their parents has it. Somebody has it, at least one of their parents has it. Somebody has it, at least one of their parents has it. Somebody has it, at least one of their parents has it. That's how you know it's a dominant. Because if it's dominant, if somebody has it, what that means is they have at least one big A. Well, that big A had to come from somewhere, which means one of their parents, at least one of their parents will have a big A. So that means that parent will also have the disease. Well, if they have a big A, it had to come from somewhere. So at least one of their parents has to have a big A, so that parent will have the disease. So if it's dominant, every time somebody has it, at least one parent has it. Every time somebody has it, at least one parent has it. That's a dominant trait. So in this example, she has it, at least one parent has it. She has it, at least one parent has it. He has it and she has it, at least one parent has it. That's a dominant trait. Okay, so the other type of trait is a recessive. A recessive is when somebody has the disease, what that means is they're homozygous recessive. Okay, if they do not have the disease, then they have at least one copy of the dominant. So this one is kind of the opposite of the other one, because the only way to possibly have the disease is if you're homozygous recessive. If you have at least one copy of the dominant, you do not have the disease. Now, this pedigree here also shows you something else that actually normally you won't see, so you can try to ignore this, but just so you know, this one is actually telling you which ones are the heterozygotes and which ones are the homozygous dominant. So in this case, if it's half filled in, it's heterozygote. If it's completely empty, it's homozygous dominant. And if it's filled in, it's homozygous recessive. But typically you won't see that, so kind of ignore these. Normally with a recessive, if it's filled in, it has to be two little a's. If it's not filled in, it's big A dash. And then you go on to figure out what the dashes are, just like you did with a dominant trait. Okay, so in this example, he has it, so he has to be two little a's. His brother does not have it, so his brother is big A dash. Now the other thing to note about a recessive disease is they're not very common. You don't see them very often. With one exception. And that exception is if there's a relative mating. So if there's incest going on. So we go back to your main point you're going to learn today, which is incest is bad. First, because it's nasty. And second, because you're much more likely to have a kid who's going to have severe genetic disease. Now the reason for that is because probably every single one of you, every single person right now that's listening to this, you are most likely a carrier for some recessive disease. What that means is you have one copy of the recessive, but since it's a recessive, you're not affected by it. You don't have the disease at all, because your other copy is the dominant, so you don't have the recessive disease. However, because you have a copy of this recessive, what that means is you're carrying it and you can pass it on to your kids. So if you have a recessive, if you're a heterozygote, you're called a carrier. Now, most likely every single one of you right now is a carrier for some recessive lethal disease. But it's unlikely that you and somebody else that's listening are a carrier for the same recessive disease. You're most likely carriers for different recessive diseases, unless you're related to somebody else who's listening right now. If you're related to somebody else, like your cousins or second cousins, then what that means is you're coming from the same family tree, so you're passing on that same recessive disease. So when you have relative mating, so incest, you're passing on that same recessive. So because of that, you're much more likely to have kids that have severe genetic diseases. Now, this was seen historically, and I am not at all a history person, so 1600 AD, 600 AD, it's all the same thing to me. But whatever it was, in history, in the European royal court, there ended up being a lot of royalty that had kids who were hemophiliacs. Now, hemophilia is a bloodletting disease. What that means is, from a simple paper cut, you can bleed out and die from a paper cut because you don't have the ability to clot your blood as well. So that is a lethal disease, especially back then where they didn't have the modern medical interventions we have nowadays. So it was a very bad lethal disease. Well, it turns out that hemophilia is a recessive lethal disease. So the only way to have hemophilia is if you are homozygous recessive. Okay, well, the European royal court, they ended up getting a lot of royalty that had kids that were hemophiliacs. The reason is because back then, if you were a prince, you could only marry a princess, and there weren't that many princes and princesses around, so what you ended up doing is you ended up marrying your cousins. So since you're coming from the same family tree, you're passing on that same recessive lethal disease, so you ended up having a lot of kids that were hemophiliacs, and we saw this historically. So again, incest is bad. Okay, so here is an example of a recessive trait. Okay, this trait here, attached earlobes. Having this earlobe shape, attached earlobes, the only way you can have that is if you're homozygous recessive. If you're homozygous dominant or heterozygous, then you do not have attached earlobes. You have free earlobes, like this earlobe shape right here. All right. Now, I'm willing to bet that a bunch of you right now just checked your earlobes. And some of you probably saw that you had attached earlobes. Okay, if you have attached earlobes, that does not mean your mom and dad were brother and sister. Okay, when I said that the recessives show up mostly when you see incest, that's if it's something that's going to kill you. Okay, something that's a lethal disease. Okay, having attached earlobes is not bad for you, so natural selection does not select against it. So we have a high percentage of this in our population. So you can very easily have people that have attached earlobes. Okay, so in this case, if you're filled in, if you have this recessive trait, then you're homozygous recessive. If you're not filled in, then you're either homozygous dominant or heterozygous. All right, so this is a recessive. Now, with the dominant, I said I could tell you it's a dominant trait, or you could have to figure it out by looking at the pedigree. With dominant, every single time somebody has it, at least one of their parents must have it. Okay, so recessive, again, I could tell you it's recessive, or you could have to figure it out by looking at the pedigree. So a recessive, the way you know it's recessive is if somebody has it when neither parent has it. That's a recessive. Because you can have two parents that are carriers, so they don't have the trait, but since they both have a copy of the recessive, they can pass it on to their kids. So that is a recessive. The recessive happens when you have somebody that has it when neither parent has it. So somebody that has it when neither parent has it. Now, it doesn't mean that you can't have it and have a parent who has it. What it means is if you ever find that somebody has it when neither of their parents has it, then that is definitely a recessive trait. So now it's going to be time for that paper and pencil again. So what I want you to do is I want you to look at this pedigree, and I want you to figure out every single person's genotype on this pedigree. Okay, so in this example, I'm telling you this is a dominant. Okay, so this is for widow's peak. Widow's peak is a dominant. So if you have widow's peak, then you have at least one copy of the dominant. If you don't have widow's peak, then you're homozygous recessive. So go through and fill this out. So pause the video right now. Fill this out. After you've completely filled everybody out and you think you have the answers, then unpause it. All right, so since this is a dominant, the way I would solve it is start by filling in the information that's easiest to know. The thing that's easiest to know is if somebody does not have it, if they're not filled in, then they cannot have a copy of the dominant, because if they had a copy of the dominant, then they would be filled in. So anybody that's not filled in, what I'm going to do is I'm going to go through and I'm going to put two lowercase w's. So that tells me that they are homozygous recessive. They don't have widow's peak. So I'm going to put two little w's, two little w's, two little w's, two little w's, and two little w's. Okay, the next thing that's easiest to know is if somebody's filled in, they have the trait, which means they have at least one copy of the dominant. So what I'm going to go through is everybody that's filled in, I'm going to put at least one big W and a dash, because it could be big W or big W little w. So big W dash, big W dash. I'm going to do that for all of the filled in ones. At least one big W and a dash. Now, the last step is to figure out what these dashes are. Now, again, you can do that by looking at either somebody's parents or somebody's kids. For example, this father here, he's filled in, so he has widow's peak, so he has at least one dominant. Now, how we can figure out what the dash is, since we don't know about his parents, is we can look at his kids. And we can figure that out because of his daughter here. His daughter has two little w's. Well, one little w had to come from mom. The other little w then had to come from dad. So we now know that he's a heterozygote. Similarly, this man here, we don't know his kids, okay, but we can look at his parents. He has a big W that had to come from his dad because his dad is a big W. His mom has two little W's. His mom had to give him something, which means his mom had to pass on to him a little W. Okay. Same for these two. These two parents have a son that has two little W's. Well, one little W came from dad. The other little W came from mom. Okay, now these two are a little tricky, okay, because they don't have any kids. Remember, these are not their kids. These are their nieces and nephews, so you cannot look at them. So you can only look at their parents. Well, let's say that he, for example, got this big W from his dad. Well, then that means his mom can give him a big W or a little w, and there's no way of knowing which one he got. So you have to leave him as big W dash. And the exact same thing for her. She got the big W from her dad. Mom could give a big or a little. There's no way to know, so you have to leave her as big W dash. So for these two, we cannot figure out what the dash is. So you could either write that as big W dash, or you could write it as big, or big, little. Okay, so that's how you solve this pedigree. Now, I'm going to want you to do the same thing. I want you to figure out this pedigree. Now, of note, this is now a recessive, which means you solve it differently. For recessive, if they have the disease, that means they cannot have a copy of the dominant. They have to be homozygous recessive. If they don't have the disease, they have at least one copy of the dominant. All right, so go through and figure this out. Pause the video right now, and then when you think you have the whole thing figured out, unpause it. Okay, so this one is for attached versus free earlobes. Okay, so what we're going to do is we're going to go through and label this. So the attached earlobes is a recessive trait. So what we're going to do is anybody who has attached earlobes, okay, they have only the recessive. They cannot have the dominant. So we're going to go through and put like 2 lowercase f's, okay, for homozygous recessive. So we're going to do that for all of the recessive ones. Then, anybody who's filled in has to have at least one copy of the dominant. So we're going to go through and put capital F and dash for those. So big F dash, do that for all of the empty circular squares. Because we know they have at least one big F, and then we'll have to go back and figure out what the dashes are, just like we did for the dominant. Okay, and then the last step is to figure out what the dashes are. Okay, so for example, these two here, they have a daughter here that has 2 little f's. Well, one little f had to come from her dad. The other little f had to come from her mom. Okay, and these two, the exact same situation. They have a son who has 2 little f's. Well, one little f came from dad. The other little f came from mom. She has a daughter who has 2 little f's, which means she had to give 1 little f to her daughter, so she has to have a little f. Because since she has 2 little f's, one came from dad, one came from mom. Now, the one that's tricky is this one here. Let's say she got the big F from her dad. Then her mom could give a big or a little f, and there's no way to know. So she has to be left as big F dash. All right, so and that's how you would solve that pedigree. Okay, so if you have a recessive disease or a recessive trait, then the only way you can have the disease or the trait is if you're a homozygous recessive. Okay, if you're a homozygous dominant or a heterozygous, you do not have the disease or the trait. So in this case, if you're a heterozygote, then you're called a carrier. Recall, as I mentioned, what that means is you are carrying one copy of the recessive. So you don't have the disease or the trait, but you're carrying it and you can pass it on to your kids. All right, so here's an example of something that's not a disease, it's a trait, which is this form of albinism. So this individual, she's albino because she's homozygous recessive. Her sister is not albino, but her sister happens to be a carrier, which means she could pass it on to her kids. Now, as I mentioned earlier, if it's a homozygous recessive that's actually a disease, something that's going to kill you, then it's pretty rare in the human population, okay, with one exception, and that's if there's a relative mating. So again, incest is bad, first because it's nasty, and second because you're much more likely to have kids that have a severe genetic disorder. So here's an example of a recessive disease that is lethal, cystic fibrosis. Cystic fibrosis is the most common lethal genetic disease in the United States. It affects 1 out of every 2,500 people. Now, and this is a disease that is lethal, will cause an early painful death. Now, if somebody has cystic fibrosis, that does not mean that their mom and dad were brother and sister. Okay, the reason these rates are so low, the reason it's only 1 out of every 2,500 is because people are not mating with their relatives. If people were mating with their relatives, then these rates would be much higher than this. And here's another example of a recessive disease that can be lethal, sickle cell disease. It affects 1 out of every 400. Now, this is a much higher rate than you would expect. But again, it is not because people are mating with their relatives. Okay, the reason this rate is higher than you would expect is because there's actually a time where it's better to be a carrier, to be heterozygous, than it is to be homozygous dominant. All right, so let me explain that to you. If you are homozygous recessive, then you have sickle cell disease, and you can die from that. So since the sickle cell allele can lead to lethality, natural selection should select against it, so we should not see the rates being this high. The reason the rates are this high is because there's a time where it's better to be this, the carrier, than it is to be this, to not have the copy of the sickle cell allele at all. And that's if you live in an area where there's a high incidence of malaria. So, if you're homozygous recessive, you have sickle cell disease and you can die from that. If you're homozygous dominant, you do not have sickle cell disease, but you can die from malaria. If you're a heterozygote, you're not going to die from sickle cell disease and you're not going to die from malaria. So there's a time where there's an advantage to having a copy of this sickle cell allele, where it's an advantage to be a heterozygote or a carrier, and that's if you live in an area where there's a high degree of malaria. So that is why the rate is much higher than you would expect. It's not because people are mating with their relatives. Now, some human genetic diseases are caused by a dominant allele. Now, a dominant allele that is actually a cause of a lethal disease is very, very rare in the human population. Why? Because if you have at least one copy of the dominant, okay, you're dead because it's lethal. And if you're dead, you can't pass it on to your kids because you're dead. So it's extremely rare to have a dominant lethal disease in humans. In fact, if somebody has this dominant lethal disease, what happened is it typically arose by random mutation. So the DNA does get mutated, it gets changed randomly. That's just a natural process that happens. We'll talk in a later chapter about how that can happen. And that's how you can get a dominant lethal disease when neither of your parents has it. All right, so here's an example of not a genetic disease, just a genetic trait. Okay, this form of dwarfism. Okay, this individual has this form of dwarfism because he has a copy of the dominant. Anyone who does not have this form of dwarfism is homozygous recessive. Now, some diseases have a much higher rate than you would expect, and that's because of when the symptoms of the disease show up. For example, Huntington's disease will cause early, very painful death, and it occurs in the population more often than we would expect. The reason it occurs in the population more often than we expect is because there are no symptoms at all until you're about 35 to 40 years old. Okay, so what that means is most people are going to have children. They usually start having children before they're 40 years old. So they didn't know they had Huntington's disease, and they passed it on to their kids. And that's why it has a much higher incidence than you would expect. All right, and we'll come back to Huntington's in a minute. Many diseases like heart disease, diabetes, alcoholism, mental illness, and cancer have both genetic and environmental components. So it's not just your genes, but also your environment that affects your chance of getting these diseases. For example, cancer. So there's lots of different types of cancer, different ways of getting cancer, but one example of your genes affecting your cancer rate is if you have a certain version of the BRCA1 gene, if you have a certain allele of that BRCA1 gene, then you have like more than a 90% chance of getting breast cancer at some time in your life. So obviously, your genes can affect your cancer rate, like that example of the BRCA1 gene in breast cancer. But it's not just your genes that affect your chance of getting cancer. It's also your environment. For example, as most people realize, if you're smoking 2 packs of cigarettes a day, you have a really good chance that at some point in your life you're going to get lung cancer. So it's not just your genes, but also your environment that affects your chance of getting a lot of these diseases. Now, let's say you wanted to know, if you have a certain disease, or what's your chance of having a disease, or what's your chance of passing on a certain disease to your kids. What you could do is you could go to a genetic counselor, and they can give you information about your chance of getting disease. For example, let's say that you have this vague recollection that somewhere back in your family tree, somebody told you that one of your relatives had cystic fibrosis. And cystic fibrosis is a recessive lethal disease. And so you're worried about that because you want to start planning a family, and you're worried you might be a carrier for that recessive lethal disease. So you go to a genetic counselor, and the genetic counselor will test you. And let's say the genetic counselor says, yes, you are a carrier for cystic fibrosis. You're carrying that recessive lethal allele. And then they'll test your partner. And let's say your partner turns out to also be a carrier for cystic fibrosis. Then the genetic counselor will tell you if you two choose to have kids, then there's a 25% chance that your kids are going to die a very painful, very early death due to having cystic fibrosis. And then you can decide whether you want to take that chance, or maybe you want to adopt or not have kids instead. Now, a lot of the genetic disease nowadays we have tests for. You can easily go out and get tested today to see if you're a carrier for cystic fibrosis. Or, let's say you want to know about yourself. Like, you could go out and get tested today to see if you have that version of the BRCA1 allele that says you have a 90% chance you're going to get breast cancer. Or, for example, Huntington's disease. Every single one of you listening right now, you either have Huntington's disease or you do not have Huntington's disease. It's one or the other. You either have it or you don't. But all of you, except for me, don't know if you have it yet. Okay, I'm too old. Okay, I don't have any symptoms of Huntington's disease, so I know I don't have it. Okay, but you might have Huntington's disease. Now, you could go out and you could get tested today for Huntington's disease. And if it turns out that you have Huntington's disease, then they can tell you that. And then you could know that, 10, 15 years from now, you're going to die a very painful early death. Or you could rather live in blissful ignorance and not know that you have it. That's a very personal decision, but you can get tested for it. So a lot of genetic diseases we can test for nowadays in an individual. We can also test at the level of the fetus. Through amniocentesis or chronic villus sampling, we can test for certain genetic diseases. For example, if we do a karyotype, remember that's an ordered display of the chromosomes from the cell of an organism. And let's say in this karyotype, this fetus happens to have 3 copies of chromosome 21 instead of 2 copies. In that case, then you know that your child is going to be born with Down syndrome. And you can start to prepare to have a child that's going to have Down syndrome. Or some genetic diseases can very easily be tested after birth. And in fact, some of them are automatically tested, at least if you're born in the hospital in the United States. For example, every single baby that's born in the hospital in the United States is automatically tested for, and I'm going to mispronounce this, for phenylketonuria. Okay, I'm going to call that PKU from now on. Okay, so every baby that's born in the United States is automatically tested for PKU. Okay, if your baby has PKU and you feed them the wrong food, then they will have severe developmental disorders. If your baby has PKU and you do not feed them the wrong food, they'll develop completely normally. So since it's very easy to test and it's very easy to treat, every single baby that's born in the United States is automatically tested for it. And then if your baby has PKU, the doctor will simply tell you, all right, these are the foods your baby can eat and these are the foods your baby cannot eat. And everyone wants to know, like, what can they cannot eat? So they can't eat phenylalanine, which is one of the amino acids. And everyone wants to know, well, what has phenylalanine in it? One thing I know has it in it, because it honestly looks scary, is if you look at a lot of those like diet sodas, on the side of that can of your diet soda, there's a big red warning label that says phenylketonuria, caution, this contains phenylalanine. And you might be thinking, why am I about to drink something that has a big red warning label on it? It might freak you out. Now, I'm not saying that diet soda is good for you to drink, but that warning doesn't apply to you. It applies to people who have PKU. So they shouldn't be drinking that. Now, personally, I think it's a separate issue why you're feeding your baby diet soda, but that's a personal choice. But it is in other things. For example, it's in turkey, for example. So this wraps up kind of the first chapter on genetics. We're going to cover 2 chapters on genetics. Now, of note, what we're doing is we're starting to build up in complexity of genetics problems. We started with a very simple, easiest thing, which is a 4-square Punnett square. We built with the more complicated, the 16-square Punnett square, which, remember, you had to foil it to do that dihybrid cross and then fill in the 16-square Punnett square. And then we also talked about pedigrees and how to know what people are on pedigrees based upon whether they're filled in circles or squares and whether it's dominant or recessive. But as I mentioned before we started, the very beginning of this chapter is this is not enough. You cannot just listen to a lecture on genetics. What you need to be doing is you need to be sitting at home and working through practice genetics problems. Okay? Genetics is kind of like math. If you're taking a math course, in order to pass your math exam, you need to be working through practice math problems at home. The only way to pass this genetics test is to be working through practice genetics problems at home. If you're not doing that, then you're going to fail the test. Okay, so in your textbook, at the end of this chapter, there's actually lots of practice genetics problems, and the answers are in the appendix. So work through those practice genetics problems, try to solve them. Any of them that you have issues with, meet with me for my office hours, and I can work through and show you how to solve them. Okay, but don't stop there. Besides the practice problems at the end of the chapter, your Master in Biology homework should have practice genetics problems. And when you go to Master in Biology and go to the study area of Master in Biology and you do study by chapter, there should be a practice quiz and a practice test for this chapter, and they should have practice genetics problems. Okay, but also don't stop there. After you do the ones at the end of the chapter, after you do the practice quiz and practice test and study of Master in Biology, also go to your lab manual, the book you use to do the labs in class. And in Appendix B of the lab manual, there are 6 pages of practice genetics problems. And they're actually broken down by difficulty. It starts with monohybrid, then it goes to dihybrid. It will also cover things we haven't talked about yet, like an X-linked gene. Okay, that's what we're going to cover in the next chapter. But start working through the ones that I have taught to you. Work through all those ones that we've covered, like monohybrid crosses, dihybrid crosses, pedigrees. And then any of them you're not sure about, again, meet with me for office hours and I'll teach you how to solve them. Okay, you have to be doing this in order to pass the exam. You have to work through lots of practice genetics problems. So please, between now and the next lecture, work through as many practice genetics problems as you can find, and then contact me for help for the ones you're not sure how to solve. Okay, so until next time.